Good morning, everyone. Welcome back to our e-seminar series on Translational Biomedical Engineering. We have a great pleasure today to host Dr. Shrike Zhang from Department of Medicine at Harvard Medical School. So before we start, and as usual, I would like to go over a few housekeeping notes during the talk. If you have any questions, please feel free to use the Ask a Question box. Of course, you can chat with other participants and the speaker and the organizers using the chat panel. Also, we have a poll for you to share your thoughts about these e-seminar series. Please make sure that you participate in the poll and share your feedback and thoughts about this e-seminar series with us. So our next speaker is Dr. Brian Tuyuta, who is a neurosurgeon from the Department of Surgery at Queen's University. Please mark your calendar and follow us on Twitter with the handle of TransLBME to get most up-to-date information about his talk and then also the future talks in these e-seminar series. Last but not least, I would like to thank our sponsor, Montreal TransMedTech Institute, which has been with us since almost the beginning of these e-seminar series. So with that, I would like to introduce Dr. Zhang, who is also a great friend of me. I know Dr. Zhang from when I was doing a postdoc in Professor Khadim Hosseini's group at Harvard Medical School, and then I can tell you that he's very knowledgeable, and then he's doing so many cool stuff, which I'm looking forward to hearing about today. Dr. Zhang received a Bachelor of Engineering in Biomedical Engineering from Southeast University in China. He received a Master's in Biomedical Engineering from Washington University in St. Louis, and then a Ph.D. in Biomedical Engineering at Georgia Institute of Technology. Dr. Zhang is currently an assistant professor in the Department of Medicine at Harvard Medical School and is an associate bioengineer in the Division of Engineering in Medicine at the Brigham and Women's Hospital. Dr. Zhang is directing the Laboratory of Engineered Living Systems, where the research is focused on innovating medical engineering technologies, including 3D bioprinting, organs and chip, microfluidics, and bioanalysis to recreate functional tissues and their biomimetic models for applications in regenerative medicine and personalized medicine. He is an author of more than 235 peer-reviewed publications with an H-index of 64, and his scientific contributions have been recognized by more than 40 international, national, and regional awards. Thank you very much, Dr. Zhang, for accepting our invitation. We are looking forward to hearing your talk. So the virtual floor is now yours. Sorry.
All right, cool. Is that showing up?
Yes.
All right, cool. Yeah, thanks, Amos and Human for the invitation. It's very great pleasure to speak here today for about our work in the past couple of years, really on how to biofabricate tissue and organ systems for translational applications. All right, so a little background to start with. As Mohsen already mentioned, I mean, I initially came from Southeast University from China, and then got my master's in BME at WashU back long time ago, and then went to Georgia Tech for my PhD, where I basically moved with my advisor in the middle of the PhD studies, and then afterwards, then I came over to Boston and basically stayed along for the last I'd say maybe 8 years-ish already. So the lab here is pretty much doing things around different corners that are allowing engineering of human tissues and organs for different applications. And these encompass things all the way from, for example, biomaterials, all the way to biofabrication, let's say bioprinting, and to biomedical devices, and again to bioanalytical kind of tools to allow all the calculations. So then the rationale for all those translational research that we have been trying to really work on is based on two things. So one thing is that in the shorter term, we have been interested in engineering these in vitro models of human tissues and organs, right? So again, the issue there is people have been using these conventional models of human two-dimensional cell cultures and or animal models, and these are not really recapitulating how the human systems are behaving in vivo, right? So then making or leading to the results that a lot of drugs that are tested positive in these conventional models, they don't really translate to the human system, right? So then that's why in the past 10-15 years, people have been really interested in developing these so-called organ chip systems to allow high content modeling of human tissues and organs in vitro to enable better drug development. But also, you can imagine if you combine these devices with patient-derived cells, you can also start to personalize medicine by screening therapeutics according to different patients' needs. And what's more important or interesting here is that you can then link these individual organ chip devices in the way that the different organs are connected in the human system. So you can now start to look at their interactions as well, and basically not only between the organs, but also between the organs and tissues with the drug molecules that you put into the system, right? So then on the other set of things, you can also imagine all these technologies that we have been working on can also be applied to engineering human tissues and organs that are functional, that can be really placed back into human system for regeneration purpose, right? So, however, I mean, comparing to in vitro tissue modeling, this is probably taking much longer time because in this case, you have to make sure that everything that you engineer is 100% functional before you can actually put everything back into the human system. But in comparison, for tissue model engineering, usually only, let's say, 80-90% of the functionality is going to be sufficient for a lot of the applications. All right, so with those in mind, I'm going to talk primarily today about two things. So how we can really harness the bioprinting technologies and the on-chip technologies to allow fabricating tissues and organs for various translational applications, right? So because again, the thing here is that for both bioprinting and on-chip applications, they have their own advantages and disadvantages. So you can either use them alone, or you can rationally combine them together in a way that the integrated system actually allows better functions of the engineered human tissues and organs. So I'm going to give some examples of each of these in the next period. All right, so again, beginning with some of the bioreactors or these on-chip devices. So as I mentioned, there can be many different variations of these chip devices depending on the final applications, right? So again, depending on whatever translational application you're trying to apply to these devices, there are different configurations you can actually make. So you can start with, for example, a simpler device having only a single chamber where you assemble all the different layers to allow hosting of the microtissue or organ structures that you engineer for long-term perfusion culture. You can also have multiple chambers on the device to allow, for example, looking at interactions between the different organs and also the migration of the cells across different chambers, for example, right? You can further build in these biophysical cues like mechanical stretches into the system to allow recapitulating of these active motions of the tissue that are experienced in the human system for some of the tissue types, right? And you can also place all the different modules together in a way that they're connected. So then you can also start to look at all the interactions. For example, here is a earlier system that we have engineered that contains not only the organ on a chip models, but also the surrounding sensing devices and also the automation kind of operating central breadboards to allow the configuration or operation of the devices in an automated manner with multi-organ configuration, right? So these are some examples, but today I'm going to give actually one example about our recent application or innovation on engineering a distal lung model of the human system, right? So if I'm pretty sure that people are familiar with all these conventional lung-on-chip models. So these have been very, very impressive and providing a lot of the biological kind of insights into the field. But if you look at the structures, they have been mostly based on this monolayer flat membrane, right? So you have a membrane in the middle, and then you apply the cells on top or both ends of the membranes, and then you stretch this membrane to allow recapitulation of these motions during breathing. So, but if you look at all these devices and compare those with the actual human alveolar lung, they are very much different in terms of structures, right? So these are these are still flat structures, but then if you look at these alveolar lung structures, they're actually sac-like structures having all these different spheres that are interconnected to allow breathing and also exchange of gas in there. So then we have been really thinking about in the past few years how we can successfully recapitulate this distal lung structure with not only localized cellular compositions, but also the global structural relevancy to allow a better recapitulation of this critical part of the human organ in vitro, right? So then we went back to this scaffolding system that we have been working on for many, many years, which is called inverse oval scaffold. So what happens here is that you can pack or actually can initially fabricate these microspheres and then pack them together in a way that they form a lattice, right? So then once they form a lattice, you deal these particles, you can infiltrate another scaffolding material into the spatial, interstitial spaces of these microspheres. And then once you cross-link this scaffolding material, you can then selectively remove these microspheres and then leave these scaffolds, called inverse oval scaffolds, having uniformly distributed sacs, having also connecting windows, right? So depending on the materials, you can of course make these microspheres out of different diameters and out of different materials, and in any situation, you can make them in a pretty nice uniform way. So then there are a few different sizes I have to mention here. There's pore size. That's basically the size of the sacs that are making up the whole scaffolds. There are sizes of the windows. These are sizes of the windows that are collecting the adjacent microspheres or the sacs, right? And then there are also surface sizes that are basically dependent on the infiltrating solutions properties. So you can actually individually tune these different sizes depending on your fabrication process. For example, if you use different temperature particles, you can very nicely tune these microspheres or these pores of different sizes. But in any case, you're still forming this uniform pores with the templating, right? So you can also modulate these window sizes by depending on the leading temperature, right? Because you have a higher leading temperature, you can actually have larger distances or contact areas between the microspheres. So that's going to lead you to a higher or larger sizes of these pores. And you can also, of course, tune the surface pore sizes depending on the infiltrating solution property. All right, so with that in mind, we have been thinking, can you actually adapt that to engineering this distal lung model for this application, right? So we actually started by fabricating these alternate microspheres and pack them together in the lattice, and then what happens afterwards is you basically infiltrate JAMA solution into this interstitial space, right? So then if you follow crossing JAMA, you can selectively then remove the alternate microspheres with ETTA solution that's basically dissolving away the calcium ions. So with that in mind, we have been also able to seed the epithelial cells, alveolar epithelial cells into the scaffolds. And very interestingly, when you have cells in there, they can gradually grow and eventually populate entire surface of these sac-like structures, right? So that's actually giving you a lot of the similarity with actual human distal lung in our body. So these cells are able to also form a pretty nice junction protein CO1. And if you do histology, right, you can see pretty nice lining of these epithelial cells along the wall of the microspheres and forming this monolayer structure. And also you can see the connecting windows between the different sacs. That's also very similar to the actual real human distal lung. And the cells were pretty much keeping their viability over days of culture. And so the material has been based on JAMA, right? So there are two things. So one thing is that it's giving you the three-dimensionality of the structure of the distal human lung. That's providing basic structural relevancy. But also we showed that using proper concentration of gamma, you can then also largely recapitulate the mechanical property of this lung matrix, right? So this is around anywhere between 3 to 6 kilopascals, which is actually very similar to the actual human lung matrix. So this is actually something that you cannot model with conventional, for example, PDMS-based devices. So again, it's infiltration process. So you can imagine not only can you work with gamma, you can work with other materials like collagen as well, right? So depending on what material you put into the space between these microspheres to make scaffolds. And you can also modulate sizes of these sacs as well. Can go all the way from a couple 100 micrometers to as large as you want, basically. But we eventually chose 200 micrometer size of the pores, since this is pretty much the size of the actual human airway lung, like sacs in the human system. So we made a lot of comparisons between our three-dimensional model of the human distal lung with the conventional models based on 2D cultures on either gamma or PDMS. So it shows basically in our 3D model, the cellular responses were much better than 2D models, whether they're being two-dimensional gamma surface or two-dimensional PDMS surface, right? So you can see on our model, the cells were actually able to grow into confluency on these surfaces, but on 2D surfaces, these are, again, still gamma. Even on gamma, right? So the cells don't really grow into confluence. They form patch-like structures in different places. And if you compare apoptosis, for example, right? So our 2D gamma culture had much more apoptotic cells than our three-dimensional gamma scaffolding system. That's much more biomimetic in terms of structures and functions. So again, I'm not going to go to any details here, but the take-home message here is that our three-dimensional gamma culture had very much different gene expression profiles than two-dimensional cell cultures, and our 3D model was apparently much more physiologically relevant than these conventional 2D cultures, whether they're based on PDMS or gamma. Again, here, same thing. So not showing any details, but again, basically indicating that there are so many different genes that are differentially regulated in our 3D model than when you compare that to 2D and 2D PDMS and 2D gamma models, right? So indicating that three-dimensional culture in structure relevant configuration is actually very important to recapitulate the functions of these cells in vitro. All right, so then what can you do, right? So you can imagine, as I mentioned, you can actually combine these biofabricated like distal lung models with on-chip configurations, right? So you can place these scaffolding systems into bioreactors, and from there you can not only deliver the medium through the bottom, but also you can start to stretch these scaffolding systems in a cyclic way, right? So that's going to give you the chance to model the human distal lung breathing event, right? And looks like the breathing is not causing any damage to the cells, and instead, it's actually promoting the expression of these junction proteins between the cells, indicating functional improvement with this motion event. And why is that important, right? So if you have a conventional model that's two-dimensional, whether it's gamma-based or PDMS-based or whatever substrate-based, even if you subject that to a breathing event, the airflow pattern in the whole device will be totally random, and that's totally not physiologically relevant. But if you compare the airflows in our model that's three-dimensional, that's very much mimicking the human distal lung, you can see a much better trajectory of the airflows within the sacs that's actually much more relevant to the human distal lung airflow when you breathe, right? So then with that in mind, you can then start to look at all the different effects of the different stimulus, right? For example, you can subject these models to smoking, and you can not only see how smoking is going to lower the viability of the cells, but also how smoking is able to cause oxidation of lipids in the cells, and also a lot of the changes in the biomarker secretion levels, right? So another thing that's interesting that we did was to apply these alveolar lung models into COVID research, right? So we first did some staining for ACE2 receptor and shows that these epithelial cells are able to express all these ACE2 markers that are that's essential for COVID, like SARS-CoV-2 viral entry. And when we applied these COVID, like SARS-CoV-2 pseudoviral particles onto these models, you can see how they were able to infect these cells on the surface of these sacs, right? So then you can also start to explore how the different pharmaceutical compounds are going to eliminate the COVID kind of symptoms, right? So and these results are actually very translational. So as you may know, hydrochlorine has been shown that has been shown to be less effective than the other molecules that have been tested here, right? So clinically, so these are remdesivir and AQD. They have been shown recently to be much more effective than HCQ clinically in these patients. So indicating that our model can potentially be used in the future for registering patient responses to antiviral drug molecules in terms of COVID and also other potential additional like outbreaks of the pandemics in the future. All right, so lastly on this section, so we also did some preliminary experiments on modeling lung cancer on a chip within this like engineered sac-like scaffolding system, right? So when you now seed the scaffolding system with lung cancer cells, you can also see how the cells are able to populate these sacs within days and stay alive over there, right? So confocal images also shows the cells were able to populate pretty much entire surface of the scaffolds. And what's more interesting here is the histology. You can clearly see how the cells in this case were forming multiple layers than a single layer that's otherwise observed in healthy epithelial cells, right? Because these are cancer cells, they don't have contact inhibition. So basically providing a chance to really model the pathology of the lung down the future as well, including lung cancer. All right, so that's the initial part. So then the second part is going back to bioprinting or biofabrication. So then there have been many different types of biofabrication methods that are available, right? So you can use its pore scaffolds for for seeding scaffolds and allow three-dimensional cell cultures. For tissue emulation, you can do fiber knitting, right? So Wilson has done a lot of things, for example, on knitting these fibers into three-dimensional structures for tissue engineering and tissue model engineering purposes. You can also have this self-assembly directed methods to allow larger scale fabrication of tissues and organs. But one of the limitations with these technologies is that a lot of times you have to do a manual or at least semi-manual way for fabricating these structures, right? So that's why in the past 10, 15 years, about 10 years primarily, 3D bioprinting has been really coming into the play of the whole field because it's a fully automation process that allows to deposit or pattern cells and biomaterials in 3 dimensions at potentially larger scale, right? So you can imagine if you fabricate tens or hundreds of these copies of tissues or organs, you can pretty much ensure the quality across different batches, right? So that's potentially much better than when you fabricate individual scaffolds in a manual way. So within the whole bioprinting field, we're having really interesting essentially every single aspect of bioprinting. So all the way from hardware engineering to software engineering and to bio-ink formulations and to downstream applications. So here are some examples of what I've been primarily working on in the lab here. So for example, we have been working on multi-material bioprinting. So rationale for that is, I mean, this is actually very important because the tissues and organs are complex, having multiple cell types coming together and multiple ECM types coming together, right? So the ability to really bring in the different cell populations and different ECM populations in there will be very essential to engineer these three-dimensional functional tissues. So we have been really having a pretty good, decent focus on engineering multi-material bioprinting formulations or configurations to allow that to happen. So then another point or focus is candid tissue bioprinting. So basically bioprinting of these hollow tissues that are widely present in the human system, right? For example, vasculature and many of the other hollow tissues. So that's also one of the key focuses in the lab here. And Bioink is another focus. So as I mentioned, Bioink is actually very important because not only has to allow in the first place the bioprinting process to happen, but also after bioprinting, how the can allow functionality and of the tissues and cells within this bioinks will be also very important, right? So within like to that end, we have been really working on different formulations of bioinks and also different processing methods for bioinks to allow proper bioprinting, but also the proper functionality of the cells post bioprinting. All right, so the other thing we have been working on is minimum invasive bioprinting delivery, basically things like in situ bioprinting to allow direct bioprinting of the tissues on the wound surfaces, right? So I'm not going to talk about this part today, but that's also something that we have been extensively working on as well right now. So yeah, so some examples of different aspects in the next part of the talk. So again, in the first place, some examples of the multi-material bioprinting. So this is actually one of the earlier works that we did back when was with Ali, right? So this is a rapid multi-material extrusion bioprinting method. Basically, in this case, you combine different outlets of the bioink formulations into a single nozzle. So this is basically a multi-channel single nozzle configuration. So the rationale for that, or the advantage of this, is when you switch bioinks in this configuration, you don't have to switch nozzles, right? So because all the bioinks are coming from the single nozzle, but from different channels. So meaning that your switching processes can be very fast. Unlike in conventional sense where you have a multi-nozzle system, you have to physically switch nozzle when you change to a different bioink, right? So that can actually slow down the process. So with that in mind, then you can start to produce more complex structures of tissues and organs in a much faster manner than the conventional multi-nozzle based platforms. All right. So then that's based on extrusion bioprinting. So that's this is another bioprinting formula configuration that we have been working on quite extensively lately, which is called DLP bioprinting, digital-like processing, right? So this is actually very different from extrusion bioprinting. In this case, what happens there is that you have a vat at the bottom which is filled with photoactive bioink. So in this case, you basically shine light patterns in a layer-by-layer manner to cross this resin or bioink in a layer-by-layer manner, right? So you're crossing one layer, lift this up a little bit, expose another layer, and crossing again, right? So if you do this repeatedly, eventually you can imagine you can basically fabricate a three-dimensional structure in a pretty nice manner, right? So this is actually giving you a much better chance to fabricate complex tissue and organ structures than the conventional extrusion bioprinting. But I mean, in the conventional sense, the issue with that is you can only have one reservoir, meaning that you can only do a single material bioprinting. Of course, you can do this manually by changing different reservoirs for the bioprinting process, but then it's again becoming a slow process. So then with that in mind, we have been working on a technology where we incorporated these microfluidic device into the vat or the reservoir over there to allow automated infusion of different bioinks into the reservoir to allow multi-material DLP bioprinting in a rapid manner, right? So then with that, you can start to fabricate these two-dimensional, three-dimensional structures of different colors or different materials, different cell types, right? So that's giving you a chance to do multi-material DLP bioprinting that's allowing more complex structures that can be fabricated. All right, so then coming back to the candidate tissue bioprinting. So as I mentioned, taking vasculature as one example, right? So vessels are essentially one of the most important components of the human system, just because almost there's no tissue organ that does not have an interconnected network of the vessels, right? So one of the conventional ways to generate vessels is called sacrificial bioprinting, which I'm pretty sure that everyone knows about it. So in this case, you basically print a little fiber that's serving as a template, and then you cast or print another gel construct surrounding this fiber and cross-link that. So once that's cross-linked, you can select and remove this fiber from the matrix to allow formation of the channels that are embedded into the gel-like structure. So again, this is hollow structures. You can basically model the structure of the vessels, right? So you can perfuse the entire structure very nicely. But also, since it's a hydrogel environment, you can then also cellularize this microchannel to allow biological function as well. For example, you can have these endothelial cells lining on the surfaces of the microchannels to allow them to go confluent. You can create straight channels or branching channels as needed, right? So that's giving you quite some flexibility. And if you culture this for long enough, again, it's because you have a biocompatible and the bioactive matrix surrounding this microchannel, right? So these endothelial cells can also then spread out into surrounding matrix to form this multi-scale vasculature, right? So this is macro vessel that you initially produce with bioprinting, but also you can form this micro vessels with spontaneous sprouting of endothelial cells in the surrounding matrix. So that's something also quite interesting. So, but then coming back, so what happened here is that with conventional sacrificial bioprinting, you're basically working at larger scale for these vessels, right? So the original scale of a couple hundred micrometers. So with another sacrificial template that was produced with mu bioprinting or mount expanding writing bioprinting, right? You can actually start with a smaller template to begin with, right? For example, these fibers are in the range of 30-40 micrometers, and if you use these as templates to begin with, you can form this microchannel that are in the range of 30-40 micrometers after removing these sacrificial templates, right? So from this microvascular network. So a step forward, you can actually even go down to single capillary levels by using shrinkage method that we have developed in the past couple of years. So basically, if you start with these 30-40 micrometer microchannel network, you can actually shrink the whole thing down to about 10 micrometers in size for this microchannels. So this is actually pretty much close to the single capillary level of the size that you may want to engineer, right? So of course, I mean, how to get cells in there is another question that we have been trying to work on right now. But theoretically, you can actually produce these very small microchannels that are close to capillaries within these hydrogel devices to model some of the capillary structures in vitro. All right. So yeah, so another interesting thing on sacrificial bioprinting is conventionally you can of course create the channels for providing the vasculature for oxygenation and all the nutrients in there. There also have been some other efforts on trying to incorporate oxygen-releasing biomaterials to allow tissue oxygenation, right? So conventionally, people have been relying on these passive release of oxygen from biomaterials. So the issue with that is since these are passive, you cannot release the oxygen for a longer time, right? So if you like encapsulate all these chemicals inside a gel, oxygen is going to go away pretty rapidly, right? So we have been thinking, can you actually provide a way to sustain the delivery or release of oxygen within the bioengineered tissues to allow long-term high-level oxygenation within the tissues to allow to basically promote tissue generation, right? So the way that we achieved this was to bioprint these algae structures that are embedded into the constructs to allow sustained oxygenation, right? Because again, these are living cells that are allowing oxidation upon photosynthesis, right? So that can give you basically as long as oxygen as you want within the tissue. So by basically modulating or optimizing the formulation of the bioinks, you can pretty nicely print these 3D structures of complex patterns, right? So these are structures without algae, and on the right, you can see when you have algae in there, these are turning green, right? So these are same structures, but then having algae in there. So adding algae is not basically affecting the bioprinting of the structures. So then if you compare or look at the viability of this algae algae cells within the bioprinted structures, you can see that the cells were pretty much alive, right? So no cells were dead, essentially. I mean, or very few cells were dead, essentially, right? So they were able to maintain their viability over time. And if you culture them at the different temperature levels, I mean, 25 is basically where the algae are experiencing normally, right? But we also show that even at 37 degrees, they are also growing over time. So it's not really affecting their growth over time. And you can also see the oxygen bubbles that are released by these algae containing bioprinted patterns, right? So indicating that you can form pretty nice oxygen kind of tension within this bioprinted structures. All right. So then, as I mentioned, these are based on photosynthesis for algae to release oxygen, right? So they can be very precisely controlled by the light that you provide. So for example, if you uniformly shine light from side of the algae within the JAMA, right? So it's going to, the algae is going to more or less uniformly grow across the entire length. But in this case, on the right, if you only shine light from top, then it's only the growth of algae toward the top, right? So you can nicely arrange the growth of algae by shining light. But also what's more interesting is that if you turn light on and off cyclically, right, you can also precisely control release of oxygen, right, within these devices on and off very precisely, right? So that's potentially giving you a chance to engineer how much oxygen can be released by this algae according to the tissue type that you are trying to engineer surrounding these algae patterns. So then next step is, of course, I mean, here we're trying to engineer tissues eventually, human tissues eventually, right? So in this case, we then embedded this bioprinted algae structure into a matrix of JAMA that's containing liver cells. So the algae cells are autofluorescence in red, and you can see the pre-labeled human liver cells in the surrounding matrix in blue, right? So you can see pretty nice co-culture of these algae cells with the human liver cells, and we don't see any issue within this co-culture system in our initial demonstration. So then if you look at then the viability, for example, comparing to control where the viability actually dropped quite a lot, the liver construct with algae in there that's releasing oxygen had much better viability, right, over days of culture. And if you look at H1F alpha expression, then without the algae, then the liver cells were also expressing much more IF1 alpha due to the hypoxia that's happening within the tissue. So then we look at functionality as well. So again, you can stand for, let's say, affecting for morphology, but also if you can basically stand cytochromes for the actual functionality for the liver cells. And with all those, you can also appreciate how adding the algae with oxygenation was able to promote the functionality of these liver cells in the co-culture system than like comparing to when you have only control without any algae in there. So another quite interesting notion here is that, again, if you're talking about eventual usage either for in vitro applications or for in vivo applications, you don't want to have algae in there, right? So we designed Bioengineer in a way that these are sacrificial as well. So this is based on primarily nanocellulose, which can be actually selectively removed by enzyme, right? So we showed that if you apply enzyme to this gel-embedded bioprinted algae structure, you can selectively remove this bioprinted algae structure quite nicely over hours, right, to form microchannels that are basically perfusable. So not only can you remove these cellulose Bioengineer with enzyme, but also enzyme is at the same time dissolving away the algae cells as well, because again, the algae cell walls are made of cellulose, right? So then you're making sure that everything's going away without leaving anything that can potentially interfere with the downstream applications. So once you have those algae removed to form the microchannels, you can then also endocellularize these microchannels with endothelial cells, right? So it's eventually becoming the endocellularized liver tissue that's having very high density of liver cells that cannot be typically achieved with conventional bioprinting or fabrication methods, right? So of course, the endothelial cells will also expressing their nice biomarkers and the CD31 and other things as well. All right, cool. So that's the symbiotic bioprinting that we have been demonstrated. So and then we have been talking about engineering of healthy tissues or healthy vessels. So you can also engineer, for example, like diseased models of vasculature, right? So in this case, we actually infuse human whole blood into the microchannel that are endocellularized, that are bioprinted, and then if you clot this blood in there, you can form actually thrombosis into these channels, right? So if you compare histology of our model, which is on the left, to actual human thrombosis, you can actually see their high similarity, right? So in our model, you have the endothelial cells that are lining in the on the interface. You have the matrix on the in the surrounding area, and you have the aggregated red blood cells in the looming area, right? So then that's actually very relevant to the real human thrombus they can find in the human blood vessel, right? Giving a chance to really model this type of disease and also look at, for example, effects of thrombolytic agents. So not only vasculature, as I mentioned, there are also other types of hollow structure that are present in the human system, right? For example, here we were trying to model breast cancer that are originating from the mammary ducts in the human breast, right? So once you see these breast cancer cells into the bioprinted hollow channels, you can see how the breast cancer cells were proliferating, but also once they became confident, how they were able to invade into surrounding matrix to form invasion, right? So basically transitioning from this DCIS, ductal carcinoma in situ, to the IDC, invasive ductal carcinoma like invasion process. And we did show that with confocal image, not only the cells were able to invade into surrounding matrix, but also they were showing this inward aggregation into the microchannel itself, right? So this is actually very similar to the in vivo configuration because, I mean, in the actual configuration, the cells not only go outside into the surrounding matrix, but also they form the lungs inside the mammary ducts. So again, indicating the like physiological or pathological relevancy of these models. So the cells were also able to deposit their own basement membranes, for example, collagen type 4 during culture, and looks like these invading cells were having more expressions of inadherent than adherent, indicating potentially this AM transition process. So a couple of years ago, we kind of worked together with this company, Exos, which is a space company, to carry over some of our bioprinted breast cancer models onto a suborbital flight. I mean, it was a very initial demonstration, but potentially gives us a chance to, in the future, look at how the microgravity or other space conditions like radiation are affecting breast cancer cell girls' invasion and also their responses to potential therapeutic candidates to benefit life both in space and also on the Earth. All right, so those are based on hydrogels. So they have been really effective in modeling the tissues or allowing generation of functional tissues for regenerative purpose. But also there are two issues with hydrogels. One thing is that they are usually more expensive depending on cell type, I mean the gel type that you use. But also on the same time, once you have this gel structure made, you cannot store them for a long time because they are subjected to degradation, right? So then that's why we have also been thinking, can you actually create a 3D printed device that's allowing cell culture or creation of these tissue models using a low-cost device that can be also stored on the shelf for longer time. So we naturally decided about paper devices. So we optimize all the conditions for printing into paper devices to allow eventual formation of these paper structures with embedded complex patterns, right, like the channels inside, which is sometimes difficult to generate with conventional fabrication methods. So then with that device, you can then basically store the device for as long as you want in the dry state on the shelf. So once you want to use these devices, you can just autoclave to first sterilize the whole device, but also second, to allow rehydration of the device. So now you can start to put cells on top or inside the channels to allow generation of the so-called bedside point of care organ chip paper devices to allow potentially personalized therapeutic screening. All right, so that's an overview. I'm not going to go into details for that for today. So another thing in the last probably 5 to 10 minutes, I'm going to talk about is so far I've been talking about those sacrificially bioprinted structures that are containing vasculature, but more vasculature embedded into tissue matrix, right? So these are basically having tissues surrounding this vasculature and the channels within the tissues. So there are also times where people may want to engineer or bioprint these standalone vessels or standalone tissues, hollow tissues for different purposes, right? So for doing that, we have been designing this multi-layered coaxial needle device for microfluidic bioprinting. So here, if you extrude the bio-ink from middle layer and the cross-linking agent from both interior and exterior layer, you can imagine a single step. You can print these hollow structures or hollow fibers that are mimicking the vessels or other hollow tissues that are present in the human system. And these are standalone fibers, right? So different than the like hydrogel embedded microchannels. And these are biocompatible inks again, so you can have cells in there as well. So after culture, you can eventually form a biologically functional standalone hollow tissue with this type of bioprinting method. So here are some examples. I mean, you can print, for example, a 10-layer structure in this case, and the entire 10-layer structure is actually perfusible, right? Because it's made of a single fiber that's been printed. And more interestingly, if you start to mismatch things, then you can create tortuosity or convolution into these microfibers, right? For example, if you perfect match bio-ink extrusion speed versus the nozzle moving speed, you can have all these nice strip fibers when you print. But if you start to increase your bio-ink delivery speed a little bit, then your nozzle moving speed. So now you're creating all those curvatures into the vessel they can print, right? And if you further mismatch that, you can build in more convolutions into these microfibers you can produce for modeling certain tissue types of interest. And believe it or not, these curvature, printed hollow fibers with curvatures, they can still be perfused. This is again a multi-layer bioprinted structure, as an entire structure is actually perfusible with liquid or air. Another potential variation is I've been showing the capacity to print these hollow perfusible blood vessel-like structures, right? So the entire vessel, the entire fiber is actually perfusible with liquid or medium. But we have also shown recently that you can print these single-end blinded lymphatic vessel-like vessels with this hollow fiber bioprinting method by basically tuning the bioprinting parameters. So you can basically form this single-end blinded lymphatic vessel-like structures of different sizes, of different wall thicknesses, depending on what you want to model, right? So, and you can imagine if you create a chip device that's having, let's say, cancer cells in between, and then a hollow perfusible blood vessel on one side, and then the other side is the bioprinted hollow or single end blinded lymphatic vessel. You can start to create microcirculation within this cancer on chip device, right? So you can perfuse, for example, molecules of drugs in the blood vessel, but then on the other side, you can start to drain the liquid from the interstitial space, which is actually something that lymphatic vessels do in the actual tissues, right? So it looks like if you compare one channel versus two channel configurations, one channel meaning that you only have the blood vessels that's delivering the agent, two channels meaning that you have a blood vessel that's delivering agent, but also the other lymphatic vessel that's draining liquid. You have very different transport profiles of the same molecules, right? So with the continuous draining, you have a little more molecules that are coming into the circulation, microcirculation. So what can that do, right? So if you, again, as I mentioned, embed breast cancer cells into the in between the two vessels, you can actually start to look at their interactions with drug molecules. So again, we first show that 2D and 3D cultures, they have differences. I mean, this is something that people all know. But also, if you compare one channel configuration with two channel configurations, they are also different in terms of cell viability, right? So the responses of the cells to the drugs, because again, the microcirculation locally are very different. And that's going to really create a differential kind of dosing of the drug to the cells in between these two fibers. So again, this is to show by doing this, you can potentially allow better stabilization of the actual microenvironment that's otherwise not possible with conventional single vessel formulation. All right, so going back to multi-materials, you can also combine these hollow microfiber bioprinting with multi-material bioprinting, right? So you can longitudinally coat the fibers basically having single fiber with different regions composed of different materials, right? So you can pattern them horizontally or vertically depending on the application. But also, you can naturally coat these fibers. So again, the rationale for this is in the actual human vessels, they're not uniform. So coming from, for example, the arterioles, they're multi-layer structure having endothelial cells in the middle and then the smooth muscle cells on that side. And going back to the when going to the capillaries, it's becoming a single layer structure, right? Only having endothelial cells, and coming back to the venules, again, it's becoming a multi-layer structure having 2 layers of cells in different locations. So how can you recreate this transitional configuration would be also something very critical to model their functions in vitro, right? So by basically turning this nozzle kind of configuration, you can start to now digitally code how many layers of walls you can build into this whole device, right? For example, you can start with 2 materials or 2 layers, and then at some point you can turn off the outer layer, so it's becoming a single layer bioprinting of the hollow fibers. And at some other point you can then turn on the outer layer again, right? So again, it's becoming a multi-layer fiber structure. So so then that's going to give you a chance to really model this transitional structure between a single layer and multiple layers depending on the actual need. So a few examples, I mean, with this configuration, you can then start to look at engineering of more complex connective tissues that are having more than a single layer, right? For example, you can print the urothelial cells in the middle layer and then the smooth muscle cells on the outside to form this ureter-like structure. And of course, for blood vessels, you can print endothelial cells in the middle and then smooth muscle cells on the outside to model this multi-layered blood vessel structure. All right, so I think finally, I also want to briefly mention, so again, I mentioned this sacrificial and shrinking bioprinting in earlier on, but you can also use shrinking bioprinting in this case. So basically, if you start with a larger hollow fiber that you can print, so this is pretty much the limit you can print with direct microfluidic bioprinting, right? So about like in the size of about a couple hundred micrometers. So it can't really go small in the conventional sense, but with shrinking, right? So you print something and then shrink that to a small size, you can actually achieve a very small size that's relevant to some of the small hollow tissues in the human system, right? So after shrinking, for example, we can achieve inner diameter of down to about 30 to 40 micrometers, which is again becoming physiologically relevant to some certain tissue types, right? So and this size is totally something that you cannot achieve with direct printing with this technique. All right, so maybe a couple final minutes. I'm going to briefly talk about these pore-forming bioinks. So again, as mentioned, bioinks are very important for our research and also for the entire bioprinting kind of scenario. So one of the interesting bioinks that we have been recently engineered is this so-called pore-forming JAMA bioinks. So it's based on a two-phase accuracy emulsion technique where you can have the JAMA forming the backbone and then PEO droplets forming this interconnecting phases, right? So what happens here is that JAMA is fully cross-linkable, meaning that you can cross-link the JAMA continuous phase after bioprinting, but then the PEO is going to remain as droplets. So you can remove these droplets from the JAMA phase to allow pore formation after bioprinting. So that's going to give you a lot of the advantages. So the reason for that is now you can control different levels of porosities, right? So again, the JAMA itself is giving you this mesh size that's around nanometer scale, but this pore-forming bioinks is actually going to give you this microscale pores to allow cellular functions. But then with bioprinting, you can create another layer of porosity, which is micropores, to allow a collective shape control, right? So this is giving you a chance to really control these different layers of pores that are important for different parts of the applications, right? So some examples, not going to go into details, but these pore-forming structures are going to actually give you much better transport of oxygen, right? For example, if you have a pore-forming hydrogel in between the two layers, you can see much better diffusion of the oxygen across this layer. But also, if you drop liquid on top, the dye molecule is going to also pass through much faster than when you drop a dye molecule onto a standardized JAMA hydrogel without any micropores, right? So giving you the chance to allow better transport of oxygen and nutrients. But also, if you look at cell interactions with pore-forming hydrogel, you have much better cell spreading than conventional standardized JAMA without any micropores, but only nanopores, right? And mechanical property-wise, that's also interesting because if you have conventional standardized JAMA without any micropores, they're easily cracked when you compress. But now, if you have a porous hydrogel, it can have much better mechanical property to allow cyclic compression and potentially injection as well, right? So, and coming back to that, we also showed how important it is to have these hierarchical pores within the bioprinted structures, right? So only with macropores it's not sufficient, right? So standardized geomer, it can still be easily cracked during injection. But if you have both macropores and micropores, then it's becoming much better in terms of injection there, right? So with 0% PEO, there's no micropores, they break. With 10, a little better. With 30, much better, right? So then they don't really break after injection. So again, showing these combination kind of effects of different levels of pores that are built into this bioprinted structure for minimally invasive applications. All right, so I think with that, I'm going to end by thanking all the wonderful collaborators and also the funding agencies, and of course all the great lab members of lab across the past years. Thank you very much.
Thank you so much. Impressive talk. I really enjoyed, and as always, lots of applications, lots of new technologies, and that's why our audience also are excited. They ask many questions in the Q&A box. I encourage, if they have other questions, put their question in the ask a question box. So I'll go through the questions of audience now. The first audience asking is, I have a question. Can microfluidic chip sent into the body be powered by the body's natural temperature?
So can a chip be controlled?
Can a microchip, which will be sent to the body, be powered by the body's natural temperature? Something, temperature responsive chip.
Yeah, potentially, yeah. So depending on the application, and there are certain temperature responsive modules you can build into chip device to be allowing temperature responsiveness, and then that's going to provide you some additional energy to power the chip for different applications. So I think that's totally possible. Although I think it's dependent really on specific application you're trying to work with the device for.
Great, thank you. Mariam is asking, the previous question was from Ali. Now Mariam is asking, thank you for your nice talk. How do you compare the sacrificial bioprinting or coaxial bioprinting to make the vessel in hydrogel? How do you think about using microchannels and oxygen releasing materials together in bioprinted tissues?
So the first question is comparing between this the sacrificial and the, I guess, hollow bioprinting. So exactly. I think I wouldn't say there's a strict, advantage of each of them. So I think really it's dependent on the final application, right? For example, as I mentioned, for the sacrificial bioprinting, it's more creating these tissue embedded vasculature. So if you're trying to, let's say, create a muscle tissue that's having these microvasculature built in, then I think that's definitely something of interest for use. But on the other side, this microfluidic bioprinting gives you the chance to really engineer these standalone vessels or hollow tissue. So, for example, if you're trying to print a larger vessel that's usually standalone, then I think that's a much better option. So I think it really depends on the final applications of the technology. You can choose things differently. So the second question was on combining oxygenation with microchannels. Yeah, so that's something that we have been doing as well. So as I mentioned in this particular paper that we just published actually early this year. So it's sort of combination, right? So we have printed these algae latent structures initially as the template to release oxygen, but then we're removing them eventually when you reach a decent density of the human cells to allow formation of the vasculature. So that's kind of sequential process, but you can also imagine, you can potentially print, you're supposed to gather in the same configuration, right? So having algae somewhere and also having the hollow microchannels built into the system. So potentially that's going to give you a synergistic, oxygen or nutrient delivery within the system. So yeah, that maybe gives you a little better, performances. But we have not tried that, so we don't know. But I assume that it's going to get something interesting as well.
Thank you so much, Dr. Zhang. I really like these cartoon drawings of your lab. But if you want, you can stop sharing so we can see each other and you can see us also.
Sure.
We can, yeah. So Raj is asking, how does the body on chip help in a space research? Any research on that line from your lab?
Well, not from our lab right now, but I'm in the US along list. So there are quite some projects that have been funded for like the so-called tissue in space, right? So tissue chips in space. So basically, people have been working with like SpaceX and the space station to basically bring some of the chip devices into the space station to allow operations over there and look at, again, how the space conditions can allow or change the behaviors of the different microtissues or organ types in there. So potentially can, well, the two things, right? So one thing, you can potentially derive additional biological, kind of fundamental biological mechanistic understanding of the process, whole, let's say, development biology and all that kind of process. But also, you can potentially look at how those extreme conditions may allow expedited development of drug molecules and also other therapeutic agents potentially benefiting the life on the Earth, right? So there have been some efforts going on for actual space research. So yeah, we're trying to get there. So.
Thank you so much. Yes, it's a very exciting field, and I follow people, especially in US and with NASA. They are doing a great job. And also SpaceX. Yeah, so Karen is asking, regarding the disease modeling example, are you manipulating the channels to create blood clots in the thrombosis on chip, or is this induced with other method?
So I'm trying to understand. So the clots are. Can you repeat the question, please?
Yeah. So she's asking basically, are you manipulating the channels to create blood clots,
okay.
or it just kind of, or is this induced with other methods?
Okay. Yeah. So I think in this case, in this particular publication, we didn't look at, for example, how the geometry or let's say damages of endothelial cells and stuff were actually inducing clot formation within the channel. So it's more an artificial kind of scenario where we actually clotted the blood in the channel. So again, that was a very initial demonstration, but I don't see any issue preventing people from trying to study, for example, how you have different geometries, sizes, or tortuosity or whatever things of the channels that are going to also affect formation of the class within the channels. Yeah, so that's totally doable.
Thank you so much. There's a question from Ishan. Great talk. Have you or anyone else tried modeling immune microenvironment responses to breast cancer or other disease using your model system, such as understanding why and how hot solid tumors become cold quickly?
Mm-hmm. Yeah, so I don't think in our system in particular, but I know there are many people doing similar things. For example, with organ chip system, for example, people have been working on well, I think this is actually a very hot topic right now, right? So people are trying to really start to look at how you can build the immune components into not only tumor, but also many of the tissue types. Because again, immune cells are one of the integral components of essentially any of the tissues, right? So that's something that people have been really doing recently as well. And I've seen people, for example, doing this on the tumor cells as well, looking at, for example, immunotherapy and all those kind of effects with immune competent tumor-on-chip devices. So I don't know anyone has done anything specific to our specific kind of system, but yeah, I know there are many research going on right now.
Great, thank you so much. Ali Raza is asking, we are usually using stem cells for regeneration of the function of the tissue as well as structural integrity of the original organ or tissue. My question is about the preservation of the stem cell, like cryopreservation. After doing the process of cryopreservation, we want to rewarm or regain our preserved stem cells. Would preserved stem cells have their original metabolic process like before cryopreservation process? This was not part of your talk, but I think it's a general question.
Sure. Yeah, I think it's more general, and I mean, I have to say I'm not an expert on cryopreservation, but from my understanding, is people cryopreserve stem cells all the time. So I assume that it's okay for cell to maintain the functions in there.
Exactly. Thank you so much. So as a follow-up question, he's asking, what would be the age of organ after doing cryopreservation of the cells? Is the age of organ the same like before?
So again,
yeah,
but I think so. It's like if you freeze something down, right? So then everything stops over there. So I don't think there's any metabolic activities going on. So I can, well, I think it's rational to assume that the same age as before it's frozen.
Exactly. Karen has a follow-up question. What software do you use for 3D models of your microfluidic chips?
Well, usually it's AutoCAD, right? So you can basically, well, depending on, again, there are different ways, right? So you can do soft lithography, so that's usually based on AutoCAD for the masks. You can also do, let's say, laser cutting if you're trying to create more larger structures. So that can be AutoCAD or CARD or whatever things that are compatible with the hardware system.
Great. Mariam has a follow-up question about the vasculature that you actually print. Why do you print coiled vascular bed, not a straight one?
Well, again, the thing is that, as I mentioned, it's not only for vasculature, right? So it's basically bioprinting of hollow tissues that are widely present in human systems. Of course, vasculature is one of the key examples because, I mean, they're pretty much everywhere. But you can imagine, you can also, there are also certain tissues that are convoluted, right? So not only, not necessarily vessels, but certain other tissues that are convoluted that are potentially giving a chance to use that kind of technique where you have all the curved structures coming together. And also, I know in vasculature, there are some disorders where you have clump vasculature that are highly convoluted too, right? So I mean, again, not necessarily like healthy vessels, but can be also diseased forms or other types of hollow tissue that are potentially convoluted in the human system.
Great, perfect. So Ruhin has a very general question about extracellular matrix. He says that the ECM plays an important role in determining cell fate. How can we incorporate ECM in organ on chip?
Yeah, ECM. So there's a ECM called decellurized ECM that's being actually quite extensively being researched right now. So especially in the last past couple of years. So I think well, we can basically derive this ECM from different tissue types, and those are becoming then tissue specific. So I think, really being able to have this decellularized ECM in combination with specific cell types for modeling different tissues can be something I think quite interesting down the road for yes, as mentioned, I mean, for matching the specific matrix properties of the target tissues.
Great. And I think this is the last question from our audience, and then I'll pass to Mohsen that has many questions. Azmi asked, great talk concerning the coaxial bioprinting or multi-coated microchannels. How can we assure that the layers are well connected with each other? And how can we control the thickness of the layers?
Yeah, so we haven't been having issues with the layer adhesion. So these are very similar materials. So for example, it's usually based on JAMA arginate and maybe some packing there depending on the application. But these are very similar materials, even if they're in the different layers, right? So then if you subject them to same cross-linking methods, their adhesions are usually quite stable over time, even if they're in different layers of the configuration. So there are different ways to control the sizes of these fibers. So basically, for example, you can control the nozzle sizes to begin with, right? So they have different sizes of different layers. So then you can easily control the thicknesses of the walls or different layers or the core or whatever parameters. But also another thing that yeah, I'm pretty sure that this is commonly done as well. So you can control the flow rates of different layers too, right? So that's going to potentially give you a chance to allow controller of the different layer thicknesses as well.
Thank you so much, Dr. Zhang. Now it's Mohsen turn. Please go ahead, Mohsen.
Sure. Thank you very much, Dr. Zhang, for your very nice and impressive talk. So each time I listen to your talk, you have something new to show. So it's very interesting and very impressive. And I agree with Human. The graphics that you used were very different, and I really enjoyed them. And then we're going to do the same. So it's very, very, very interesting. So I have a few questions about some of the work that you did on algae cells, and then because this is the first time I was at least listening to this talk, to this part of your talks, and then my question is about the metabolic competition between the algae cells and then some of the cells that are in the tissues. For example, liver cells are highly metabolic. And then do you see any issues in terms of the metabolic competition, in terms of like the consumption of the nutrients that are present in the media between the cells that you're growing and then the algae cells that are there and they're producing?
Yeah, well, I mean, in our initial demonstration, we didn't see that. So the cells that are co-cultured together were I guess both pretty happy in there. So that's one thing that we observed. So yeah, but I can imagine. So we have some following up projects where we did see some competitions over there. So I mean, one cell type is potentially growing much faster than the other cell type. So then that's why where the problem comes. But yeah, but again, I mean, this is very first demonstration, and a lot of things we still don't know. So, but I mean, from the theoretical point of view, I mean, there should be a symbiotic relationship over there, right? Because again, oxygen can be released by the algae for the mammalian cells, but also the mammalian cells are going to release, let's say, CO2 and also the other metabolites that are potentially taken up by the algae cells for their metabolism. So theoretically, but again, I mean, a lot of things practically we don't know yet. So we're still working on understanding a lot of the symbiotic kind of relations over there with some follow-up projects. So hopefully, yeah, next time I'm going to give you much better answers for this.
That could be very interesting to understand, especially with all these studies being done on metabolic interventions, and then the interactions between the cancer cells and then the immune system, and then also the stroma cells. That could be a very interesting study that can add to the to these research field in the area of cancer research, and then also other types of tissue engineering areas. So my other question is about the work that you did on genetic profiling of the cells that are grown on PDMS, and then you compared them with GELMA 2D and then also GELMA 3D. Did you take the effect of matrix stiffness into consideration? Did you consider that effect?
Yeah, so that's
studies?
why we actually had different configurations in there, right? Because we not only had PDMS, but also we had 2D JAMA for comparison as well. So for 2D JAMA, then the matrix stiffness is pretty much the same as the three-dimensional kind of structure. So then but even there we saw a lot of differences between gene expression. I mean, in terms of gene expressions between the two configurations. So yeah, we can rationally assume that, I mean, it's both the structure and also the matrix stiffness that are affecting how the cells are responding to these microenvironments. So yeah, so yeah, that's becoming again important when like consideration when you're trying to model these tissues in vitro.
So I'm going to ask you a very different question now. It's not mostly scientific. So I'll ask you two questions. The first question is about all these technologies that you're developing. So I noticed that you are developing many different platform technologies, and then you're patenting them. What are your plans for bringing them into market. These are, each of them, they can be a startup company. I mean, you've noticed that some of these technologies, there are companies that that are doing similar things, and then you bring something new to the field, and there is a potential there. What are your plans to commercialize this technology?
Mohsen, they need you to put your cash down and then bring those in the market.
I was waiting for that day. So,
No,
yeah,
but I mean, I think.
Yeah, I mean, it's definitely. I mean, that's something that we've been thinking about, but it takes time, as you know, right? So, again, I mean, not only time but effort. So, again, I mean, so. Yeah, I mean, getting all these things done in the lab is already taking a lot of efforts. So we haven't been getting really too much time to really, bringing anything into the actual market. Although some things are kind of undergoing in place right now. So there are some discussions with people or other companies on potential licensing opportunities. But I mean, they take time.
Yeah,
so,
it takes time. Yeah. I agree. So speaking of time. I know that you have a daughter who has this almost the same age as my daughter, almost 3-4 years. How do you manage to do all this cool stuff and then still spend time with your family? I need to know your secret. I don't know. I mean, this is something that I always wanted to ask you, and now it's the time.
Not only that, Dr. Zhang is a great photographer, and he spends time in nature and going camping, climbing, I don't know, and he's a traveler and the stuff. Like, this is a very, very relevant question.
I have a story that I want to tell. But
sure. So I guess, I mean, first thing is, since it's going to be recorded and then made public, so again, family is important, right? So, again, I mean, it's a lot of the help from family. I mean, they're taking care of the kid and a lot of other things where, like when I'm not around, right? So, it's certainly important. But also on the other side, I guess it's different things, right? So I think sometimes switching your mindset is actually pretty like facilitating the actual efficiencies, right? So if you're just like staying here for the whole time, then your brain gets a little kind of very, I guess, a little. I don't know what's the correct word over there, but I mean, you feel tired and you're finishing the jobs, right? But if you, let's see, once in a while turn to something else, refresh your minds a little bit, then things get better. So but again, I mean, I wish I had more time to go birding and do all the things. Limited time, but still, I mean, even that, I mean, even once in a while go for birding or for traveling. I mean, again, last year has been really tricky, right? Because there's no travel, but usually I go, as you know, I mean, I go for traveling pretty frequently. So I think that's actually very refreshing. So even traveling can be quite tiring as well, but it's different types of tiredness, right? So then I mean, your mindset is actually different. So I think that's actually very helpful in terms of promoting efficiency when you're actually working.
So I know, I mean, because I know Dr. Zhang very closely, and I know that how much he spends with his family, and then it's very admiring to see that he's not only spending time on these cool projects, but he's also spending time with his family, and he's making sure that he spends good quality time with his daughter and then his wife, and then so he's a role model for many of us. And then the story that I wanted to tell was that about a few years ago, before COVID, Dr. Zhang was in Victoria. And then we went out for just a quick hike. And then he's so great in, listening to the nature and then distinguishing the sounds of different birds. That was pretty amazing. So he knows birds. It's like the way he prints stuff. He knows also birds too, a lot. His knowledge about birds is as much as his knowledge about bioprinting. And it's very interesting to see. Again, I mean, the birds were very far away, like it was like hundreds of meters far away, but he was able to listen to them and then tell me what, a bird is making this sound. So thank you very much, actually, Dr. Zhang, for accepting our invitation. And Human, I don't have any other questions.
I just want to thank Dr. Zhang, and looking forward to catch up in Montreal or Victoria.
Sure, yeah.
And soon. And just want to encourage the audience to follow up. We have next week a great speaker, Dr. Brian Toyota from Queen's University. He's a neurosurgeon, and he's talking about his research and his translational research that he's doing. Same time, same place. Thank you so much. Take care of yourself.
All right, thank you.
Take care. Stay safe. Bye.
Sarah.